Okay, so I think we can get started this afternoon. For those people that don't know me, my name is Maria Strack, and I'm the president of the Biogeosciences section at CGU. Welcome to the second Biogeosciences session of this CGU online for the summer. And we have 4 speakers today. But before we start, I just want to remind you that the AGM, the CGU level AGM is following this meeting starting at 3:45. So we do encourage you to stick around for that. And we also have a couple other Biogeosciences events coming up in the next couple weeks. So next week is a poster session where there will be a bunch of Biogeosciences posters. And then August 21st will be our section AGM at the same time, as well as the student awards. So we also encourage you to join us for that. So we're going to get started today. Just a couple notes are that if you do need closed captioning, that was shared in the chat. If you have questions for the presenters when they're done, you can put those into the Q&A, which is should be at the bottom of your Zoom window, and then I will read those to the presenters. And each of the presenters will give a talk of about 10 to 12 minutes, and then we'll have some time afterwards for questions. So our first presenter today is Sean Leipe, and I'll turn it over to him to share a screen and introduce his topic.
All right, thanks, Maria. All right, so can we see my screen here? We're good?
Yep, that looks great.
All right, so thanks for tuning in today. My name is Sean, and I'm here to talk about my thesis work at McMaster with Dr. Sean Carey on landscape scale variation of vegetation change in the Yukon. Now, just before we get started, I'd like to thank the Global Water Futures and Northern Water Futures program for the funding that made this work possible, and the McMaster Watershed Hydrology Group for all their help and support over these past couple years in and out of the field. Now, most of us probably know there's been a global increase in temperature over the past 40 years that disproportionately affects high latitude ecosystems, and this is expected to continue into the foreseeable future. A somewhat underestimated consequence of this is an increase in vegetation across the Pan-Arctic, which we can see in the green and blue on this figure up here, which has been documented by large-scale remote sensing, as well as plot-level studies. A major component of these changes is shrub expansion in Arctic and subarctic ecotones. Now, the rates of shrub expansion are highly variable depending on plant species, topographic position, hydrology, soils, and other ecosystem properties. Due to vegetation's first-order controls on water, energy, and carbon balances, understanding how these rates vary across the landscape is critical for the future of northern watersheds. So as an example, increased rainfall interception and snowmelt from shrub canopies causes increased runoff, which leads to changes in the water balance. Carbon and nitrogen cycling rates are also enhanced under shrub canopies due to reduced erosion. And the expansion and densification of shrubs decreases albedo and increases the absorption of solar radiation, which can lead to a positive global warming feedback. Now, shrub expansion has been estimated using larger and medium-scale satellite greenness indices, not smaller scales using historical air photos and plot studies, but there's limited research on shrub change detection with LiDAR. And just to give everyone an idea of what these northern shrubs can look like, here's our field researcher, Dave Barrett, who I think is about 6'3", standing next to some of the larger ones in our study area. Now, this study's main goal of comparing changes in vegetation was split up into 3 main subcomponents. First, we wanted to see if we could estimate shrub heights using LiDAR accurately enough to compare changes in that over time. Since that may or may not have been feasible, looked into creating aerial coverage models based on the LiDAR for this change detection. Then, use one or both of these methods to compare how vegetation change varies across different ecosystem and physiographic variables. The study was conducted in Wolf Creek Research Basin, which is a large subarctic montane watershed about 15 kilometers south of Whitehorse. Wolf Creek's a long-term research facility with over 25 years of continuous monitoring data. And due to logistical issues caused by Wolf Creek's size and accessibility, we focused on Granger Basin here, which is a smaller headwater subcatchment. It straddles the alpine and subalpine ecozones with a large elevation range of about 1350 to 2080 meters above sea level, has a continental subarctic climate, and is dominated by dwarf birch and willow shrubs at lower elevations. Now, just quickly for anyone who doesn't have much remote sensing background, airborne laser scanning, or more commonly referred to as LiDAR, is an active remote sensing method where pulses of light are emitted from an aircraft-mounted laser instrument and reflected back to the sensor from objects on the Earth's surface. Laser pulses from these LiDAR sensors can penetrate vegetation cover, which allows us to directly measure the 3D distribution of vegetation canopies and the topography below. Let's get back to Wolf Creek here. Our 2007 LiDAR survey was flown by Dr. Chris Hopkinson and the Applied Geomatics Research Group, and 2018 was conducted by Dr. Brian Menounos at UNBC. Having two of these point cloud data sets available for the same study area gives us a great opportunity to explore changes in vegetation between survey years. For all of these objectives, in order to evaluate how well the LiDAR can model vegetation, we needed to get some boots on the ground and confirm what's actually there. This was done using vegetation transects spread across the study area to evaluate shrub properties and compare to LiDAR metrics. Here's an example of one of these transects over some UAV imagery we took in the field. Recorded the plot centroids with a differential GPS before measuring vegetation height at some predefined intervals, along with taking some percent coverage estimates and a few other measurements we're not really going to get into today. Now, one issue that needs to be accounted for before making any comparisons between years is the resolution differences between surveys. You can think of LiDAR as a sampling method in some ways. The laser beam footprint from the sensor is only going to capture a fraction of what's actually on the ground. So, if less of the plot area is sampled, LiDAR is more likely to miss the highest points of vegetation and underestimate heights compared to field measures. This was accounted for by randomly retaining a specified fraction of 2018 returns to create a new thin point cloud with the same resolution as 2007. Now, after the usual pre-processing workflow, extracted LiDAR return heights within each plot radius, which we can see here for this one transect, and compared them to field measurements. The idea here being that if the thin point cloud can still give accurate results compared to field data, could potentially evaluate changes in height between surveys through the whole study area. And to show how this whole process presents, let's look at the LiDAR returns within this transect here, same one as before. When we clip out the original 2018 height above ground returns, we can see a pretty realistic looking resolution or return distribution. And it looks pretty consistent with the field measures that we took during this study. But after thinning it down to 2007's resolution, there is going to be a pretty significant decrease in accuracy. So for these comparison plots I have here, we have LiDAR return heights on the y-axis, compared to both the maximum and average heights measured within each shrub-dominated field plot on the x-axis. And we have a dashed 1-to-1 line to compare to the regression in gray. The original 2018 LiDAR seen here only underestimates max plot heights by about 14% on average, which is pretty good compared to what previous works have found. However, we can't directly compare these to 07 yet because of the resolution differences between surveys. So let's see how the thin point cloud matches up. And as one might expect, results are a fair bit worse using this. Our squared values for both comparisons drop off the map, and our new average underestimation of about 60 centimeters is over 40% of the actual field measured heights. And we can attribute a lot of this error due to the large amount of plots without a non-ground return in it due to increased sampling error compared to only one before. Now, due to these high levels of field height underestimation, I decided to focus on estimating shrub cover and comparing changes in this instead. First step here was conducting some supervised classifications of shrub presence versus absence using machine learning algorithms in R. Predictor variables for these came from rasterized LiDAR metrics from the thin 2018 survey, and the models were trained and validated on field data from the veg surveys. After a bunch of testing, we found that the random forest algorithm gave better accuracies and stable classes than support vector machines. That was applied to 2007 and 2018 to make final classifications. Now, overall, these final layers show a 63% increase in shrub covered area from 07 to 2018, which works out to about 5.8% yearly. Now, this is a pretty large increase yearly compared to other studies on Arctic shrub expansion, which could be due to Granger Basin's higher productivity, as it's located at a lower latitude compared to other sites, which have mostly been done in northern Alaska, and it has a warmer continental subarctic climate. The increase of vegetation at this scale will have a pretty wide range of implications for water, energy, and nutrient cycling within Granger Basin. And since we know that shrub expansion rates vary across individual study areas, just measuring this by itself isn't really enough. For that purpose, created a shrub change map by subtracting the 07 layer from 2018, then separated these results into distinct change categories to explore landscape scale variation with. Next step was to derive some terrain indices to compare shrub cover changes over, which we can see looping on the right of the screen here. These included elevation, slope gradient, northness, eastness, stream distance, topographic wetness, and relative slope position. We can see some pretty obvious patterns when we look at these side by side with the shrub change map. And the first step in actually quantifying the differences is extracting terrain values from the different shrub change categories, which was done using Wilcox and rank sum tests to compare value distributions. So what these tests actually do is they'll tell you if there's a statistically significant shift between two vectors, according to an alternative hypothesis of greater or less than. So as an example result, these tests showed us that areas of increasing shrub cover have lower elevations than stable shrub, loss, overall Granger Basin, and areas with stable non-shrub in order. When comparing the expanding and stable shrub pixels to other classes, these results held for all terrain indices under consideration. So as another example, previous works have found that shrubs preferentially expand into areas with a higher topographic potential for wetness. So in this study, using the greater than alternative hypothesis, we found that the stable and expanding shrub classes had higher values of the Saga wetness index than all the other shrub change categories. All these tests showed statistical differences with p less than 0.01, except for one testing if areas of shrub gain had a lower slope than shrub loss, which had a p of about 0.49. Now these Wilcoxon tests will tell us if there is a statistically significant shift between categories. They don't really tell us much about the magnitude of these differences. So that was done by binning terrain values into 5 separate classes, then comparing the percent of pixels in each shrub category falling within them. Categorical comparisons here showed that the biggest differences between shrub change areas were found when considering overland flow distance to stream, elevation, and relative slope position. So as an example here, although only about 32% of Granger Basin is located within 500 meters of this interconnected stream network down here, a whole 67% of expanding shrub pixels are found in these areas. And the same thing applies for elevation. About 10.2% of Granger is located below 1450 meters above sea level, but 28% of expanding and 31% of stable shrub pixels were found within this elevation class. So just to wrap things up, the main conclusions we found in this study are that shrub classifications from LiDAR surveys found a large increase in cover within Granger Basin between 2007 and 2018, with a yearly rate that either matches or exceeds previous works on northern shrub expansion. These shrubs are found in and are expanding into areas of lower elevation, lower slope gradient, more stream proximity, with a higher topographic potential for wetness. Further research is required to distinguish between the different types of shrub expansion, such as infilling, increasing shrub size, and new colonization, and how these vary with topography, and to determine how the complex interactions between vegetation, soils, and snow cover impact hydrologic processes. A greater understanding of these changes and their consequences is crucial for predicting the future of northern watersheds under our rapidly changing climate. Thanks again for tuning in today, and I'd be happy to take anyone's questions.
Thanks, Sean. So if we have time for questions, so if anyone has any questions, they can add them to the Q&A box there. While we're waiting, Sean, I'll ask a question. I know you had some problems not problems, but you couldn't really look at heights because of over time, because of the lower resolution of the 2007 data. But I was wondering whether you think there's some potential for LiDAR there to also maybe map changes in biomass over time.
Oh yeah, 100%. So there's one paper, I don't have the title on hand, but it's from Heather Greaves et al. In 2016 that uses LiDAR and some optical imagery to quantify shrub biomass at a really fine scale, like 25 centimeter pixels. So if we had 2 surveys of the same resolution, that's very doable. And with more identical acquisition parameters, because it's pretty sensitive to that.
Right, that'd be really interesting, I think.
Yeah, and integration of UAV imagery or LiDAR would be pretty cool too, like drone mounted sensors.
Well, feel free to add some questions if anyone else has any. Okay, maybe oh, I lost mine. I was going to say I'll ask one more, but I lost the idea there. Are there any plans to fly the site again now? like in the future, to be able to like sort of build on that?
That decision's a little above my pay grade. That's a question for Sean Carey. It would be pretty cool to see.
It would be, yeah. Oh, I'm getting one question here. What was the cause of the shrub decrease in those areas that showed that?
So not 100% sure on that right now. So other surveys or other studies on shrub expansion have found decrease in shrub pixels. So some of that could be due to issues with the accuracy of surveys. Like we had to do this at a reasonably high resolution, so like 2-meter pixels. And so issues with that or shrub heights being missed by the thin LiDAR could have caused that. And then not 100% sure what the physical processes behind the loss would be.
Yeah, I guess further investigation into those. But I guess you didn't find any clear changes in your statistical analysis or predictors of that.
What's interesting is that the areas with shrub loss, according to these hypotheses, so we'll say stream distance, for example, like the lost pixels were closer to streams than the areas of stable non-shrub or the overall Granger Basin's distribution. So it would make sense that they're located in areas that would have been shrubs before due to higher productivity or other related process.
Right. Okay, well, thanks, Sean. I think we're going to move on now, keeping on schedule. So our next speaker today is Emily Prystupa. And Emily, I'll just have you share your screen and introduce your topic.
Hi, everyone. Just trying to get into, there we go. Everyone can see the screen?
Yeah, that looks okay. Good. Yeah.
Awesome. So hi, everyone. My name is Emily, and I'm just graduated from my master's at the University of Waterloo. And today I'm going to be presenting on some of my master's research, which is on the response of dissolved organic carbon dynamics to salinity in a constructed fen peatland in the Athabasca oil sands region. So before I go further, I just wanted to thank the organizations that funded this research, and also thank both the lab groups that I'm a part of for all their help, especially those who helped me in the field. I also wanted to acknowledge that my research takes place within the boundaries of Treaty 8, which is the traditional territory of the Dene and Cree, as well as of the Métis of northeastern Alberta. So peatlands perform a variety of ecosystem services that are very important. And one ecosystem service is carbon storage. With peatlands occupying approximately 4 million kilometers squared of land and storing an estimated 547 gigatons of carbon, this represents a very significant carbon store. However, development on peatlands may reduce these ecosystem services, and this is being done in northeastern Alberta, in the Athabasca oil sands region, due to surface oil sands mining, which has already disturbed 953 kilometers squared of land as of 2016, of which about 50% of the land prior to disturbance was peatlands. Fortunately, companies are mandated to return the land back to an equivalent land capability, and this is being tested with a pilot constructed fen. So the constructed fen is located in a larger constructed watershed, where hill slopes and an upland help to drive water movement into the fen. This water movement occurs through a tailing sand aquifer, which is high in sodium and has led to mobilization of the sodium into the fen. This sodium may affect some of the objectives set out in the fen's construction, with my research specifically looking at whether it will impact the carbon accumulating function. Specifically, my objective is to evaluate the role of sodium on DOC quantity and quality in the rooting zone of the constructed fen. So to provide a little bit more background of the importance of dissolved organic carbon, dissolved organic carbon, or DOC, can be decomposed within the site itself, contributing to on-site greenhouse gas emissions. It can also be exported downstream, where it can contribute to greenhouse gas emissions downstream, which would not be considered as part of the peatland carbon budget unless DOC export is considered. DOC can also bind to contaminants and mobilize them, potentially to downstream environments or within the fens to the vegetation. or potentially precipitating them out of solution and making them less mobile. So sodium, as I mentioned, is a large factor in the constructed fen due to the tailing sand, and sodium can impact the three main processes that affect dissolved organic carbon in the fen. These are decomposition, rhizodeposition, and solubility. So sodium can affect decomposition by affecting the microbial community, causing stress. It may also impact plants, and thus rhizodeposition, which is the release of carbon from plant roots. And finally, it can affect solubility by binding to the DOC, potentially help bridging it to precipitate it, or depending on concentrations of other ions, potentially mobilizing it into solution. So to study this objective, I installed suction lysimeters at 10 and 30 centimeters throughout the constructed fen, trying to target the different vegetation types that are common. And I sampled them biweekly, analyzing the samples for sodium concentration and DOC concentration, as well as a variety of other parameters that were expected to influence DOC dynamics. I also measured two measures of the quality of DOC using spectrophotometric properties. And these are SUVA and E2, E3. SUVA is proportional to aromaticity, and E2, E3 is inversely proportional to molecular weight. So a higher molecular weight and more aromatic compound will be harder to decompose and is considered recalcitrant compared to the lower molecular weight or less aromatic compounds, which are considered labile as they're easier to decompose. For this presentation, I'm mostly going to be focusing on E2, E3, but just for reference, the effects were generally similar between both of these measures. So now going into some results. I found that between years, DOC concentration was roughly stable. However, the quality of the DOC shifted to become more labile. Looking into what might have controlled this, I looked at the effects of sodium on DOC, and I found that as sodium concentration increases, we had an increase in DOC concentration and an increase in the E2, E3 ratio. So potential explanations for this are increasing rhizodeposition, where if you have sodium impacting the permeability of the plant roots, this will lead to a release of carbon from the roots, which is generally small, contributing to a higher E2, E3, because remember, it's inversely proportional. And from this increased release of carbon, you have an increase in DOC concentration. It could also be explained by a decrease in decomposition, where potentially the sodium is causing stress to the microbial community, preserving more labile and smaller compounds in solution, thus leading to higher concentrations. So sodium, as it has increased at the constructed fen between years, it could potentially explain this increase in E2, E3 between years. However, despite sodium being correlated with an increase in DOC concentration, we don't see this effect in the interannual trends. So I decided to look at this further using an RDA. And I found that, as expected, sodium was a large control on DOC concentration, with graminoid cover, specifically looking at sedges and rushes, also being correlated, strengthening that rhizodeposition mechanism potentially. I also found, however, I found that the quality of the DOC was largely controlled by the sulfate concentration, where higher sulfate led to a higher E2, E3 ratio, so lower molecular weight compounds. One potential explanation for this is that sulfate can help to precipitate DOC, and thus would lead to a decrease in the solubility of DOC. Since this solubility effect largely impacts the larger compounds, this would leave some of the smaller compounds in solution, but overall contribute to a lower concentration of DOC. So since both sodium and sulfate have increased at the constructed fen between years, these may explain the interannual trends, where additive effects on the quality for E2, E3 led to the DOC becoming more labile between years, whereas the opposite effects on the DOC concentration led to a lack of difference in the DOC concentration between years. So now moving into some of the effects on the temporal trend over the growing season, which the study was conducted in 2019. I found that over the course of the growing season, there was an increase in DOC concentration and a decrease in the E2, E3 ratio. This has been found at reference sites in the boreal and has generally been attributed to the increases in temperature over the course of the growing season, where when you have higher temperature, you have higher decomposition. This decomposition favors the production of DOC and preferentially removes the smaller, more aromatic compounds, leaving behind higher molecular weight compounds in solution. This effect was supported at 30 centimeters using an LME and the RDA for temperature. However, it was less clear what the effect was for 10 centimeters. So here, looking at some of the results from the RDA at 10 centimeters, I found that the model, a lot of the variability in the models was attributed to the random factor. And this random factor was the plot-specific location of where samples were taken, since they were taken at the same plot between different sampling events over the summer. So potential explanations for this high degree of variability attributed to the random factor are differences in the substrate quality between plots, where we have, where although vegetation cover was considered in the models, it didn't consider the quality of the peat or the quality of the litter. Since vegetation patterns have shifted, the litter may be different than the surface vegetation cover. Additionally, there could be local drainage conditions where an area of local drainage may be aerated and have higher decomposition than some of the other areas at the site. So overall, I found that at 10 centimeters, DOC was spatially variable, and this was potentially due to the variable substrate for decomposition or local drainage conditions. At 30 centimeters, I found DOC was controlled by sodium, and this was likely due to an increase in rhizodeposition or a decrease in decomposition, where sulfate was also identified as an important factor contributing to DOC dynamics at 30 centimeters. As sodium levels are expected to continue to rise in the constructed fen, we may expect to see increases in labile DOC. And as labile DOC is more mobile, it may increase carbon export and contribute to mobilization of heavy metals at the site. So therefore, I recommend that industry should measure DOC export for consideration in the carbon budget of the site and assess the role of DOC in metal transport. Thank you everyone for listening, and let me know if you have any questions.
Thanks, Emily. And yeah, if anyone has any questions for Emily, please add them to the Q&A. Actually, I have one question here, Emily. It's from Carl Mitchell, and he's wondering about the sulfate links. Do you know much about what happens to the sulfate in the system? Does it contribute to acidity at all? Do you smell a lot of sulfide and know the sulfate is getting reduced a lot?
So there is very high amounts of sulfate on the order of 1 gram per liter. So most of the dynamics that I would expect would be due to, for DOC specifically, would be due to solubility instead of the reduction pathway. There are some areas in the site where you can smell the sulfide when the water has been sitting for a while, but it's not as much as you would really think based on the high level of sulfate. But it is clear that it is being reduced. And then in terms of the acidity, one thing that's interesting is that the pH of the site is around 7. It's around neutral, and that's another thing where there's so many ions and going on in the system that it's very difficult to figure out. It's not like the sulfate is one dominating over a bunch of other ones. It's just very high concentrations of ions throughout the site. I'm not sure if that answers the question. Yeah. Okay.
Yeah, it does. And I mean, I believe there's quite a bit of carbonate in that site too. Carbonates and bicarbonates.
Yeah.
So that probably complicates things a little more. I had a question that I was wondering, you had some hypotheses there about precipitation, and is there a way that you would be able to investigate that DOC that is precipitated?
Yeah.
Like how much of it is there?
Yeah, so there's kind of two possible ways. There's potentially diluting the sample or using a dispersing agent to be able to get at some of that DOC. You could also measure, it wouldn't tell you the precipitated fraction specifically, but if you measured TOC, you could get an idea under different things. But also you can use different filter sizes to assess the different fractions under different sizes, and potentially using a slightly larger than the 0.45 micron standard, you could get precipitated, but not TOC, but precipitated DOC.
So yeah, small-ish, but getting caught on the filter. Yeah. So we have another question here from Graham Clark, who's asking, did your LME include seasonality or time of year? And if not, could that be why the random site-specific variance was so large?
So the LME didn't include the time of year for that specific LME because I used the random factor as the plot location. However, I also, while I was doing the stats for it, I did consider models that did use the seasonality, and for that it was attributed to the trends and not as a adding it into the LME with all the other factors that I was including just complicated the variance so much because it was divided between too many things. So I had to select the models that were best. So I used stepwise selection.
And did you say you had temperature in those models that might be a good indicator of seasonality?
Yes, and temperature and water level as well, because the water level tended to rise over the course of the season. So temperature and water level were included. And the temperature was averaged and not point specific, but was taken about twice a week using a temperature curve and averaged.
Well, thanks, Emily. I think in the interest of staying on time, we'll move on to the next talk. So I'll just get you to stop sharing your screen, Emily.
Yes, of course.
So our next speaker is Linh Trần, and I'll just have Linh share her screen and introduce her topic. And you're on mute still, Linh. So just
Okay. Can everyone hear me now?
Yep.
Okay, cool. I'm just gonna share my screen.
Perfect. Looks good.
Okay. Hi everyone. So my name is Linh, and today I'm gonna talk about the optical characterization of dissolved organic carbon, DOC, and the water chemistry in the wetland pond in the Perry pothole region. So this is my proposal presentation. We have some data from the pilot study last year, but mainly I'm gonna talk about what I'm doing right now experimentally, and then what are my plans for the future. So let's talk about wetland first. Wetland, even though it occupies a small portion of Earth's landscape, is a very productive ecosystem. Broadly speaking, there are two types of wetland. There are the coastal wetland and the inland wetland. The Perry pothole wetland region belongs to the freshwater inland wetland, and it's mostly in the upper of Midwest US and then the Western Canada. We call it pothole because during spring, the rain and the snow melt gonna fill in the low-lying area from concentric body of water, as you can see here on the right side, and that is the pothole. The hydrology of pothole vary a lot, depend on the location and the season, or even from pothole to pothole in a given wetland type. So in wetland, there are a lot of nutrients and they interact with each other constantly. As a member of a mercury lab, we're interested in the mercury myrtle cycle in the Perry pothole region and what role of DOC actually play into that. And that is the big picture. For my role in that is I'm gonna monitor the photochemical degradation of DOC regarding space and time by sampling multiple wetland ponds and using incubation method. So the photo degradation, photolysis of DOC is a process when UV radiation gonna affect and alter the makeup of DOC. Through UV radiation exposure, DOC is going to be oxidized to become low molecular weight or even simple DOC. And then the process repeat again and again until the molecule is small enough. So it's not going to be broken down by UV anymore. But if it's small enough, it's going to be consumed by bacteria to produce CO2, CO, or sometimes even methane as the final product of DOC decomposition. Oh, and mercury is actually favor newly formed DOC, the more complex DOC, to partner with and move around. So with the change in DOC structure, we're going to see the change in DOC movement in the wetland pond, and sometimes even its ability to form methylmercury. And we want to study that process in the peripodal wetland using Sandinese National Wildlife Area as our main study site. This area is strictly for research, and it has minimal human activity, minimal tree shape, so we can maximize the UV radiation exposure, and it has a mostly dry atmosphere. The first part of my project is the seasonal collection. So I'm gonna collect DOC and measurement a lot of other characteristics like sulfate, conductivity, pH for bi-weekly for an extended period of time. And then we're gonna try to monitor the concentration of DOC to see whether it's gonna change seasonally or not. The second of my project is the incubation. So when we do an over survey of the area, we're gonna narrow down into two ponds, pond 2 and pond 118. So they have the concentration of DOC is hugely different, one almost double the other. And we're gonna take water from these two ponds, we're gonna filter them through a 0.45 micrometer glass filter to have a water sample with only DOC. Then we're gonna incubate them with one as a light incubation and one dark incubation. We're going to incubate them at the surface level at a 0.25 meter and 0.5 meter. We collect them bi-weekly for every 2 weeks up to 12 weeks. So our goal is to see if we can expose DOC with a range of UV radiation during the time that DOC tend to peak, which is summer, to see if there's any change in the component of DOC. And we're looking into the component using optical analysis. And we measure both fluorescent and absorption using the Duetta spectrometer. So we are having one of the approach is the specific UV absorption or SUVA. The SUVA is taking the absorption of DOC at 254 nanometer and divided it by the DOC concentration. It's relatively to the aromatic compound of the DOC sample and gonna tell us and they're gonna tell us about how it contributes into the DOC in general. So last year we have a pilot study where we use an incubation method, but we collect it every 3 weeks instead of 2. And in this graph, you can see here is the x-axis is a SUVA measurement, and the y-axis is a SUVA measurement, and the x-axis is the incubation time by weeks. There are some changes in SUVA that we can see, and generally the point behave kind of similarly to each other, even though they have a huge difference in DOC concentration. However, this is not enough data for us to draw any conclusion. Even though SUVA is really good to look at the aromaticity of DOC, we also want to look at a bigger picture. So that's why we're using emission excitation matrix, EEM. EEM is a type of control plot that provides you a 3D scan. As you can see here on the left side, is between the emission wavelength versus the excitation wavelength versus the fluorescent intensity. And then depend on the concentration of the component in DOC, the peak is gonna vary on the location. EM can detect a very small portion, typically in the part by billion range of the common component of DOC like amino acids, sulfuric acid, and humid. assets. There are multiple ways of using EEM. Assets provide a huge amount of data. And the one way of using it is we're gonna try to look into the molecular weight of the component. So on the right side here is one example of using EEM with a parallel factor modeling. By doing that, we can narrow down all of the components into molecular family based on its molecular weight and seeing how these molecular family gonna change over time. As you can see here, like we are in a very early stage of our project. And we have, right now I'm just focusing on the DOC data collection and my 12-week incubation to collect both seasonal and incubation sample. And then we're moving on, we're trying to analyze everything using SUVA and EEM. And we're also comparing between pond to see how the survey of the area is going to be. And we're comparing also between the incubated sample and the natural sample over time to see if there's any difference. And lastly, like I mentioned, is that we're going to link all of this into Mercury. So Mercury is going to be our main focus and see how all of this change in the optical characterization of DOC going to affect the Mercury cycle in the wetland pond. As we're not really trying to like isolate it one DOC component and try to characterize them because it's going to be massive task. So we're just trying to offer like a novel idea of how DOC and mercury behave in wetland pond and how in a bigger picture that's going to affect mercury as one of the pollutants. I want to thank Ansel and UFR giving me a chance to work on this project and everyone who have been helping me doing all of this during the summer. And I'm going to take any question if you guys have any. Thank you.
Thank you. So are there any, if there are any questions, I'm actually already seeing some coming in, but anyone else feel free to add them to the Q&A box. So this is a question from Carl Mitchell, and he's just first saying he might have missed it, sorry, but what types of mercury measurements are you going to use to look, what are you going to look into? Are you going to look at things like photodemethylation, inorganic mercury reduction, both, or other things?
So for the mercury part, let's say that is going to be a bigger picture. That's something we're going to work toward in the future. But for now, I'm focusing on DOC. Well, in order to answer that question, we're also going to look mostly, I think, in the concentration of methyl mercury comparing into if we're going to see any changes in the optical property of DOC. So it's going to depend what we're going to find in this project to determine how we're going to move forward with our mercury measurement. During the past year, our Dr. Brittle have been trying to measure the mercury and monitoring mercury concentration in the pond, so we can use that all of that data together and kind of compile like a big mercury budget of the wetland region.
Great, thanks. I had a question about your pilot study, and maybe you haven't had a chance to analyze it yet, but you sort of saw some increases in SUVA there, which I guess you would maybe anticipate the other way, maybe? Would you kind of expect a decline? And so I guess my question really is, did you see a change in the DOC concentration, or maybe you haven't seen that data yet?
So the SUVA is the absorbance of DOC at 254 nanometer divided by the DOC concentration. So it's normalized by DOC concentration. And it's really surprising that we kind of see an increase and then decrease now. And it's even more surprising that this happened in both ponds. It's not just like one pond. I kind of expect DOC to become smaller and simpler. So over time, we kind of hopefully decreasing, but it might be kind of, because let's say it's a really short period of time. So it might be some fluctuating up and down during the year, depend on the temperature and other component in the whole area. So it might be that, and that's why for this study, we're gonna do it a bit longer to see if it is gonna trend going up and down again. And then if that is the case, then we're gonna average it out to see if there's a trend that's going down. Yeah, but my hypothesis is it's gonna go down with as we expose it more into the UV radiation. Yep.
Thanks. We're a little ahead of schedule, so if anyone does have any more questions, feel free to send them along to Lynn here. Okay, just a follow-up question from Carl. Do you know if there's any iron in these systems?
Yes, I believe that there are not that much. Because iron also complex with the mercury, so we potentially gonna consider it to be one of our predictor. However, I think the concentration here is not that great, and so far we don't see a huge impact of iron on either DOC or mercury. So we don't really intensively look into iron in these ecosystem.
Okay. Yeah, so Carl's mentioning, and I think it may even affect the EEM data sometimes, but
Yeah.
Carl's mentioning sometimes it affects the SUVA. So if that's something that is regularly measured, at least that's good to know in case you need to correct for it later in your calculations.
Yeah. Yeah, we kind of at this point is because I haven't really got a chance to analyze any of my data yet. So I'm just like collect everything and to see if we can get anything out of it.
Great. Okay, well, like I said, we're a couple minutes ahead, but I think in the interest of making sure that Igor has some time for questions and that we're done by the start of the, oh, as I say that, I have one more question for you, Lynn. So we'll do this question because we're ahead of time. Okay. This from Colin Whitfield. Do you know what the dominant source of DOC is in your ponds? Is it autochthonous?
So I have the data so this is not the data from this year because again, I haven't analyzed it yet, but from last year, we see a lot of terrestrial data, terrestrial DOC compared to bacterial one. So last year I analyzed the source of DOC using fluorescent index. So I use a cutoff I think it's 0.2.7 as a cutoff to see whether it's going to be terrestrial or it's going to be bacterial, and we see they're kind of like a mix between terrestrial and bacterial, and there is not a lot of like we cannot really confirm whether the sample is going to be like a lot of bacterial or this is going to be a lot of like terrestrial DOC. So that's why I'm going to want to focus it more on the EEM and the bad effect to see if we can have a better picture of it, because bad effect also provide us a source of it depending on the peak of the DOC. But yeah, I would say that it's more it's more terrestrial DOC, but it's definitely a mix. Yeah.
Thank you. Okay, so we'll move ahead now to the last talk for today before the AGM. So remember, stick around for the AGM after we see Igor Lehnherr's talk. And Igor, I'll just let you share your screen and introduce your topic.
Thank you very much, Maria. I also like to thank and congratulate our three previous speakers for really interesting and excellent talks. I was very impressed. And thanks to Lynn for starting to transition this session from wetlands and carbon to a little bit more of a mercury angle, which is what I'm primarily going to be speaking about today. This is work that was led by my former master's student, Stephanie Vardy, describing how seasonality drives methyl mercury cycling in a high Arctic catchment. So we're going to move a little bit further north, and I realized that maybe not everybody here has a strong or a lot of background in mercury. So I'll start with a bit of an introduction. So mercury.
Igor, do you want to do, we can see like the whole PowerPoint?
Yes.
Perfect. Thank you.
Thanks. No, that's great. That's much better. So yeah, mercury is a environmental and global pollutant because of its ability to undergo long-range transport. That means that even remote areas like the Arctic are impacted by anthropogenic sources of mercury. However, those emission sources are, we think, maybe stabilizing because of things like the Minamata Convention on Mercury, which is aiming to reduce emissions around the world. So climate change is potentially the main driver of further change in the biogeochemical cycle of mercury at this point in time. And that's one of the things that we really wanted to look at with this work. The other reason why this is these kinds of questions are important is because northern indigenous people across the circumpolar Arctic are exposed to mercury and methylmercury in particular through the consumption of traditional country foods. And if we look at the this figure here at the bottom, showing that they have concentrations of mercury in their blood that is higher than what, or can be higher than what the US EPA guideline is. So there's also some concern from a human health point of view. So today I'm going to be talking a lot about methylmercury and mercury methylation. So I thought I'd introduce why these are really key concepts. Most of the mercury that's emitted by anthropogenic sources is inorganic, but it can be converted to this form called methylmercury via a microbial methylation. And this is important because methylmercury is more toxic, but also is the form of mercury that's bioaccumulated and biomagnified in food webs. So a lot of this mercury methylation can take place in anoxic environments and aquatic environments especially. So thinking about compartments like freshwater sediments, wetland soils, anoxic bottom waters of lakes, etc. So with that a bit of background, I'll state what our objectives are for this study. And we really wanted to understand what the sources of methylmercury are at the aquatic terrestrial interface in Arctic ecosystems, and what happens to methylmercury when it's produced, how it is transported downstream, what is its ultimate fate, does it get bioaccumulated into food webs, into aquatic organisms, and within that, thinking about sources, are there hot spots for mercury methylation in these types of Arctic catchments? And seasonality is another important thing that we wanted to examine because on one hand, we have a real paucity of data that's obtained from any time outside of the open water season. So we wanted to collect some measurements before ice out, before snow melt. And also because understanding seasonality and interannual variability can also help us to make better predictions about how climate change may impact mercury cycling and methylmercury sources in these types of environments. So this is to give you an overview of our study site and a visual representation, so you have a picture in mind when I present data from the different sites. Here we have our what we call our skeleton continuum, a freshwater hydrologic continuum. This picture is taken from the top of a mountain looking down. We have a system that's fed by seeps that are fed by permafrost meltwaters that flow into a lake, Skeleton Lake, a series of smaller ponds, and then into a wetland. So we have our wetland inflow site all the way down. We have a wetland outflow site, and then into a more channelized creek that eventually flows down into Lake Hazen, which is an important ecosystem in terms of also supporting an Arctic char population as well. And just to kind of give you a bit of an idea of where we are in Canada, the inset map here shows you that we're right at the top of Ellesmere Island in a location called Kitinepak National Park. So I mentioned seasonality as something that we really wanted to investigate, and I'll be presenting data contrasting spring and summer. And I just wanted to give you a bit of an idea of what I mean when I say spring, very different than many of you might think of from our experiences in temperate latitudes. So even in the springtime, we're looking at conditions like this, where we have a lot like extensive snow cover, at least 1.5 meters of ice on top of the lake water column. So these two pictures are taken from similar locations. You can use the Pingo in both photos as a way to orient yourself. So here we have Skeleton Lake, probably late June with a little bit of ice cover left. And this is, I think, a picture taken May 30th or maybe June 1st, so around that time. So we'll be contrasting the spring season where we have snow and ice cover with the summer season, which is really representative of open water conditions. And we have 2 years worth of data. 2015, which was a much warmer summer with greater thaw and greater flow through the continuum, and 2016, which was cooler and therefore less thaw and generally drier conditions. So the idea for this study was to collect samples all along this continuum, so from our thaw seep, our lake, ponds, throughout the wetland and the creek, and to try to look at where does methyl mercury, where is it produced, how is it transported downstream? And we both sampled water, soil, sediment, really anything we could think of throughout this continuum. But we also performed some methylation assays, which I won't talk about too much today, just to quantify rates of mercury methylation. So again, where and how fast is methyl mercury produced, because we really are interested primarily in methyl mercury as a more toxic and more bioaccumulative form. So this is data from the 5 main sites throughout our continuum, and showing on top our warmer 2015 year and our cooler 2016 year. And for the most part, the trends are very similar. We see very low concentrations of methyl mercury, and I should also clarify, so the dots represent concentrations of methyl mercury, unfiltered and filtered, and the bars are percent methyl mercury. So how much of all the mercury is present in this methyl mercury form, which we use typically as a proxy for mercury methylation as well. So we have very low methyl mercury concentrations coming out of the permafrost meltwaters, and the concentrations then increase as you move downstream through the lakes and the lake and ponds, and at the wetland inflow they peak and then surprisingly start to go down, and by the time we get to the creek where it flows into Lake Hazen, concentrations are once again really low, which is very interesting because from what we know, our paradigm from temperate and boreal ecosystems is that wetlands are really important sources of methyl mercury to downstream ecosystems. So we're seeing something a little bit different here where the wetland is in fact acting as a methyl mercury sink. What is interesting by comparing the 2 years is that in the warmer year, there's a much smaller loss of methyl mercury moving through the wetland than in the cooler year. So the ability of the wetland to act as a sink is also dependent on, or is weakened by warmer and wetter conditions. And I'll get into why that is, but essentially we think because warm, wet conditions favor mercury methylation. So what is happening in this wetland, which was potentially, as I said, a bit of a surprise because based on our knowledge of temperate and boreal wetlands, we would have probably expected methyl mercury to increase throughout the wetland. So one of the things, and I'll flip back to point this out, is there's probably the wetland is acting as a sink for particulate matter and suspended solids. So the concentrations of methyl mercury throughout the wetland are declining sharply, and it's primarily the particulate bound methyl mercury that's showing this. So the wetland is trapping particulate matter and thereby trapping methyl mercury, but it also is doing a lot of other things that are really interesting. So it definitely, even though it acts as a sink overall, there's a lot of mercury methylation happening, but it's only in some parts of the wetland. So here's a transect of soil cores across one section of the wetland. And what we see is that wetter organic wetland soils have much higher percent methyl mercury values. So this is where mercury methylation is happening. And then drier sites, so the yellow and red colors, drier locations along our transect, and deeper soils that are more mineral in nature are poor environments for mercury methylation. So the wetland definitely shows a lot of spatial heterogeneity as well in terms of its role on methyl mercury cycling. We think also that the soil organic matter in these wetland soils is quite good at absorbing methyl mercury as the water flows through these soils. And actually one of our strongest predictors for methyl mercury concentrations in wetland soils was soil organic matter content. It was a better predicted than, for example, measured rates of methylation. And then the other thing, so we've got so far trapping of particulate matter, absorption of methyl mercury on soil organic matter, and the third mechanism that allows the wetland to remove methyl mercury from flowing waters is demethylation, so the decomposition of methyl mercury. And this is illustrated here. This is some of our methylation assay results. So on the left panel, we have methyl mercury production, which is higher in the lake and the pond sediments and lower in the wetland soils. And then here we have degradation, which is actually is higher in the wetland than it is in the lake and pond sediments. So it illustrates that demethylation is a bit more favored in the wetland compared to the lakes and ponds. And this overall this is consistent with the spatial and temporal survey that I showed a couple slides back. So I've been talking a little bit about seasonality as well. So I'm going to switch gears now and step away from the spatial component and talk a little bit about the seasonal differences that we see. And one of the things that was really striking that we observed is that methyl mercury concentrations in the water of Skeleton Lake are pretty low in the summertime. But when we sample those same waters from under the ice in the springtime, they're about 10-fold greater, which is probably, again, another surprise, because in most instances in temperate and boreal lakes, we would expect summertime concentrations to be much greater because conditions are warmer, you have more mercury methylation happening compared to the winter ice-covered season. So we're seeing here very high concentrations of methyl mercury under the ice in the winter or in the springtime compared to the summer. However, if we look at rates of methylation in the lake sediments, they're still higher. So Km is our metric of mercury methylation, is still higher in the summertime. So what likely is happening is that the persistent ice cover is, on one hand, helping facilitate the development of anaerobic conditions or anoxic conditions which favor methylation, but also preventing ultraviolet radiation from penetrating into the water column and therefore shutting down photochemical degradation of methyl mercury, which is probably pretty important in the summertime as well. So if we do a bit of a mass balance, it turns out that over this ice-covered season, there's as much the sediment water flux of methyl mercury into the water column is on par with what we've measured in the summertime in other Arctic and boreal lakes. So again, a bit of a surprise that there's this much biogeochemical activity occurring over the course of the ice-covered season. So what does this mean for zooplankton? So despite the fact that we have really high concentrations of methyl mercury in the water under the ice, zooplankton at that same point in time are actually 5 times lower in their methyl mercury content compared to the open water season. So again, our methyl mercury concentrations are shown here. So May 28 is a sample collected under the ice, quite low zooplankton concentrations, much higher in the open water season, which is a bit of a surprise considering that the pattern in the water concentrations is actually flipped and is the opposite. So bioaccumulation factors are very low under the ice, but are actually fairly high in the open water season. So potentially what we're seeing is that in this gap of about a month and a half during ice out, there may be enough residual of these high springtime methyl mercury concentrations that then get taken up into the food web once the lake comes alive. So once the zooplankton kind of come out of their more dormant stage and start feeding and reproducing. So the final piece of the data set that I want to present pertains to snowpacks. So the top graph here shows again methyl mercury concentrations throughout the continuum, and snow methyl mercury concentrations are quite high. Again, kind of comparable to these really high under ice water concentrations I was just talking about, and higher than some of these other measurements that we made in the summertime. So lake summer waters and ponds, etc. And so this is interesting because it represents a really large reservoir because the snowpack is obviously everywhere. So at snowmelt, there's a potential for a very large release of methyl mercury to downstream ecosystems because the snowpack holds 25 times more methyl mercury than the Skeleton Lake water column. However, the source of this methyl mercury in snow is quite unclear. We did some methylation assays in snow. Methylation rates were very low and very unlikely to explain what we saw. And potentially we're thinking some of this methyl mercury in the snow could be coming from blowing soil dust because the snowpack isn't very deep, and there's some exposed slopes that sometimes can contribute to blowing dust. So just with the last couple minutes of my time, I wanted to bring this back and think about what this kind of what these data mean for our understanding of how climate change will impact methyl mercury cycling in these ecosystems. So as we saw from our comparison between our warmer and our cooler summer, warmer conditions mean more thaw, wetter wetland soils, and greater hydrologic connectivity throughout the continuum. So on the one hand, it favors methylation. So anybody who studies methylation will look at this picture and think, wow, we've got some pretty saturated soils and warmer conditions. That's probably pretty good for our microbial communities that are responsible for mercury methylation. More water flow also means greater efficiency in terms of downstream transport of methyl mercury to the Lake Hazen ecosystem and food web, because more flowing water means better transport and lower retention of suspended solids, and particulate-bound methyl mercury, and shorter residence times through some of the drier wetland soils, which are responsible for demethylation. So to summarize, in the springtime, we have these really elevated and surprisingly elevated concentrations and proportions of methyl mercury under the ice. The snowpack is also a very large reservoir of methyl mercury. In the summertime, we have through our continuum some sources of methyl mercury in the upstream portions of the continuum, but by the time this water flows into our receiving downstream water bodies, so Lake Hazen, concentrations and proportions of methyl mercury are very low for the reasons that I discussed. So a couple of takeaway messages. Lake and pond sediments and saturated wetland soil support methylation hotspots during the open water season, but they're not necessarily widespread, particularly throughout the wetland. The export of methyl mercury to downstream ecosystems is limited by mechanisms that include particle settling, binding of methyl mercury on soil organic matter, and demethylation in drier soils. And perhaps something that we may not have expected is that even in the really cold winter months and ice-covered season, we still see continued methyl mercury production in Skeleton Lake, which was very interesting. And finally, the snowpacks are large reservoirs of methyl mercury. And if we tie these pieces together and think about climate change is likely to result in both greater methyl mercury production, so greater methylation, but also more efficient downstream transport of methyl mercury and potentially greater bioaccumulation in downstream aquatic food webs, such as the Lake Hazen food web. So thank you for your attention, for sticking it out on a Friday afternoon. And I'll take any questions at this time.
Thanks for the interesting talk, and I'm sure some questions will start popping up here. Yeah. So Lynn has a question for you. Have you looked into the DOC or DOM in the water in the lakes and the wetlands?
So we have, so I should also say that some of this work we've been working at this site in collaboration with some other people, and some of whom have been a graduate student of Sherry Schiff's. And if you're familiar with Sherry's research, you'll know she was probably very interested in looking at DOM characterization and concentrations. And so we've looked at it a little bit. We definitely have some data on DOM characterization throughout the continuum, but there was nothing that really jumped out at us in terms of DOM quality being able to better explain spots where we saw more methylation versus more demethylation, for example.
Thanks. I have a question mostly because I'm not a mercury person, but what are the kinds of conditions that promote demethylation?
So so demethylation is more of an aerobic process than compared to methylation. So if you have less waterlogged soils, more oxidative processes, and exposure to light. So certainly in terms of fresh waters, if they're exposed to ultraviolet radiation, you do get quite efficient photochemical degradation of methyl mercury as well. So that's an important demethylation pathway.
Okay. So I guess if that's happening in the wetland, it's like kind of maybe well mixed water or something. Like maybe that's why the inflow has higher rates?
So the inflow is higher rates because those soils tend to be wetter for a greater proportion of the season, because they're immediately downstream of the lakes and ponds. So those soils are more saturated and kind of have the types of conditions that favor mercury methylation versus the outflow, where the wetland kind of spreads out and a much greater surface area is relatively dry,
Okay.
which would favor demethylation.
Okay, that makes sense. Okay, so I have a question from Carl Mitchell here. He says, nice talk. This question is in relation to the high methyl mercury concentration under ice. Skeleton Lake looks pretty shallow. Is the thickness of the ice pretty similar to the water depth? Just trying to get an idea if something like ion exclusion from the ice could play a role in the trends you saw. And he says, did you measure methyl mercury in the ice by any chance?
So that's an excellent question. And so the ice is about 1.5 meters thick and the lake is 4.5 meters deep. And so cryoconcentration, so this idea of exclusion of solutes from the ice during freezing, accounts for about a doubling of methyl mercury concentrations under the ice, but there's still beyond that. It's a 5-fold increase comparing open water to under ice that can't be accounted for by solute exclusion or cryoconcentration. And we have a handle on that because we can compare things like conductivity and dissolved ions to get an idea of what this kind of solute concentration we get as a result of ice formation. So the methyl mercury concentrations are really high under the ice is definitely significantly above and beyond that mechanism. We did not measure methyl mercury in the ice. We have data for the snow and the water, but not the ice, which probably would have been a good idea to get for sure.
Hindsight is 20/20, right? You can only measure so many things. Well, we're just about at the time where people are hopefully popping on for the AGM. As we kind of wait for a couple more of those people, if there's any more questions that pop up sort of in the next 15 seconds, we'll take them. Okay, seeing none, I'm going to thank again all the speakers for those excellent talks today. It's been really fun to still get a chance to see everyone's research, even though we're so far apart. So thanks again, everyone.